This meeting is being recorded.
All right, well, I'm going to go ahead and get us started. So my name is Annie Carter, and I am the program coordinator with Colorado Canyons Association. For those of you who are unfamiliar with our organization, we are a nonprofit, and we work on education, outreach, and stewardship in the 3 national conservation areas here in Western Colorado. Our organization works very closely with the Bureau of Land Management to do this public outreach, to run education programs, and to host events such as this. So we have a lecture series every year. We typically do about 3 presentations in kind of the shoulder season, spring and fall. And so this is our first virtual lecture of the fall. We're joined by Dan Kowalski, who has close ties to CCA. He's actually the husband of our education director, but also an aquatic research scientist with Colorado Parks and Wildlife. And he's going to be telling us about the salmonfly tonight and his research on that important species. If you have any questions during the presentation, we will have time for questions at the end. You can go ahead and add those questions to the Q&A box or the chat box, and I will ask those questions at the end of the presentation. So you can put those in throughout, and I will bring those up at the end. This is going to be recorded, and it will be posted on our YouTube channel probably tomorrow. So if you don't get a chance to attend the whole lecture, or you'd like to share this with someone, you'll have access to that recording. It'll be emailed to you. So with that, I'm going to go ahead and hand it over to Dan. Thank you so much, Dan.
Okay, thanks, Annie. Can you hear me?
Yep.
Okay, well, thank you, Annie, and thanks for CCA inviting me to give this presentation. Hopefully you guys will find it interesting. Today I'm going to talk about salmon flies, and about a third of the presentation will be kind of general biology and ecology of this aquatic invertebrate, and I'll try to tie it to the Gunnison River and the Gunnison Gorge NCA a little bit, and then the rest of it will be just my confused ramblings and time for question at the end. So what we're going to be talking about today is Pteronarcys californica, which is one of the largest stoneflies in North America, and specifically in Colorado. It's not only the largest body, but it's one of, in my opinion, one of the most charismatic aquatic invertebrates, if an insect can be charismatic. As we're going to hear today, it's an important part of both aquatic ecosystems, and it's important to a lot of people recreationally and even economically. You may have heard these bugs called many different things. I'll call them salmon flies. I'll call them generally stoneflies. Some people call them willow flies. They've also been called girdle bugs and even bunion bugs. In a movie in 1992 that just about ruined fly fishing, depending on who you ask, Brad Pitt's character, Paul McLean, caught a large rainbow in a pivotal scene in that movie. He caught that fish on a bunion bug, which is a cool old-timey fly pattern that was tied to to imitate a Pteronarcys stonefly. So this is the bug that we're talking about. It's Pteronarcys californica. It's a large stonefly species that lives most of its life in rivers and a small portion of its life as an adult flying around. Here's a bunion bug that I referred to earlier. This is a fly tied to imitate it, and it's really a pretty good imitation of it, considering it's from the 1920s. So we're going to begin with some of the evolutionary history of salmon flies. This is a species that is very, very old. Here you'll see on the screen a fossil member of the same family of Pteronarcys californica, and this fossil is from 165 million years ago. It's a very, very old species. So this fossil is probably 165 million years old, but the authors of the study that found it estimate that the species could be as old as Triassic or even the Permian age, 250, maybe 300 million years ago. So to give you an idea that, the earliest birds that we've identified are about 150 million years ago. And if you look at this species, the fossil, I mean, it's remarkably similar to Pteronarcys that we have today. So it's truly what people would call a living fossil. To tie it back to the Gunnison Gorge, these prehistoric stoneflies look very similar to today's salmon flies, and they were around, about the time as the oldest sedimentary rocks in the Gunnison Gorge. Really like the early to late Jurassic period. Those fossils that I showed you were from China. They weren't from North America, but it's about the same time that a lot of the geology in the Gunnison Gorge was being created. So to go into some of the biology and ecology of this bug, they are primarily plant eaters. They eat leaf litter, filamentous algae, and they're primarily eating, they do eat some other insects, but they're not carnivorous or considered carnivorous. Usually when they eat other insects, it's just as a mistake, or just when they're eating other leaf litters and filamentous algae. They're considered an indicator species of cold, clean, quality river habitat. They're very intolerant of disturbance and pollution, and basically general river alterations, from flow regime to temperature regime. Anytime rivers are messed with or polluted, generally this bug does not live in them. They're also, which is very sensitive to metal pollution, which is important in Colorado, because a lot of our rivers with historic mining influences do have some metal pollution issues. So they are one of the largest aquatic invertebrates, probably the largest aquatic invertebrate you're going to see in Colorado, other than maybe Dobson flies. And they're certainly the longest lived aquatic invertebrate. If you take nothing else away from this presentation about them, know that they have really long lives, and they attain a very large size. And when they emerge from the river to the adult form, it happens in a very short period of time. The annual emergence lasts between 5 and 13 days, and this is a very, very short emergence. Most aquatic insects have emergences that last a month or 2 months, or even throughout the summer. So to have a focused emergence that lasts, maybe a week is pretty rare. And they can dominate the biomass of invertebrates in our rivers. Because they live for 4 years, you have 3 different age classes of the bugs in the river at any one time. And couple that with their large size, and they really are a huge amount of the biomass of aquatic invertebrates in some of our rivers, like the Gunnison, the Colorado, the Rio Grande, and the North Platte. And because they're so abundant and they're so big, they're very important to sport fish, specifically trout, as a diet item. They can, a study we did on the upper Colorado River in Middle Park estimated that where they're abundant, they can be anywhere from 65 to 75% of the average annual diet of rainbow and brown trout. So they're stonefly species. They have an incomplete metamorphosis, which means they have 3 life stages: egg, larvae, and adult. Mayfly species have a complete metamorphosis. They have a pupal stage as well. As I said, it's a 4-year-long life cycle, and importantly, they have a really long diapause. And what that means is, in this portion of the life cycle right here, after the eggs are laid, those eggs live in the river sediment for somewhere around 8 to 10 months, maybe. We don't know exactly because they're kind of hard to sample, but where it has been studied, they live in the eggs lay unhatched in the sediment for 9 to 10 months before they emerge as a very small, couple millimeter long first instar larvae. Those larvae maintain that larvae form for another 3 years, shedding and growing, but generally looking the same and just getting larger and larger before 4 years later emerging as an adult insect. And importantly, these bugs don't hatch out of the water like a mayfly does. They crawl out of the water. So when the emergence occurs, the nymphs gather near the edge of the river, primarily in riffle areas, and at night they crawl out of the river, emerge from their exoskeleton, and become a flighted adult. Now, an important thing that I want to emphasize during this presentation is these bugs are not only important to people who are only interested in the river, like anglers. You know, of course, fly fishermen love the emergence of this bug. It produces some of the best fishing of the year. But these bugs are important not only to the river community, but to the terrestrial community as well. I don't want to overstate this, but in the same way that salmon take marine-derived nutrients and bring them into the forest, these bugs transmit carbon and nitrogen and phosphorus from the river into the riparian areas. And, you know, it's not on the same magnitude as maybe salmon, but when you think about a river like the Gunnison Gorge, you know, this is a desert canyon. Most of the upland areas are pretty unproductive. And so the amount of carbon and the amount of nutrients that they're transmitting out into those systems is probably significant. And we've done some work on this, and you know, there are some models that have been developed on how this carbon flux occurs, and those models were done with species that are a lot less dense and a lot smaller than Pteronarcys californica. And when you looked at the predictions of those models, we measured carbon subsidies going from the river to the terrestrial environment that were 250 times the predicted annual carbon flux of normal invertebrates. So clearly those models and those estimates that were done on other species are kind of out the window with these guys. These guys really transport a lot of carbon and a lot of nutrients from the river into the riparian areas. Here's a shot of the exuvia of this bug in the cobbles on the edge of the Colorado River. And as you can see by the density of them, it's not an insignificant thing. This is a really high biomass of insects when they do come out in high densities. And they're not always in this high density, but in rivers in Colorado, like the Rio Grande, like the Colorado, and like the Gunnison Gorge, they do occur in extremely high densities. Here's a shot of the adults. This is sort of a mating swarm that would be occurring in the willows above where all those shed exoskeletons that I just showed you are. And as you can see, it's a pretty big pile of insects and quite a bit of nutrients. Here's a picture in the Gunnison Gorge. I believe this is kind of just downstream from Chucker Rapid of the emergence, and it's a little bit hard to see all the adults. All those little light specks that you see above the river are flying adults in a pretty giant mating swarm. And this doesn't occur all the time throughout the emergence, but when it does occur, if you get to see it, it's an amazing experience. So of course, these bugs are important to anglers. And, you know, I couldn't help but include some pretty cool footage of, you know, bugs eating salmonflies on the surface of the river. This is some really neat slow motion footage that some guys were working with from Arizona shot on the Colorado River near the pump house. Pretty cool stuff and neat footage. So like I said, I mean, these bugs are important not only to the fish, but they're transmitting a lot of nutrients to terrestrial species. Here's a cool shot of a wolf spider eating them, and it's not just spiders. It's a lot of birds. Just about any bird species that lives in a river like the Gunnison Gorge is going to eat them. Garter snakes eat them. I can't prove it, but I assume ringtail cats and things like otters eat them in the Gunnison Gorge. And I think over somewhere around 20 different species of terrestrial animals have been documented eating these insects. And it's not just animals. This is a neat study from California that they documented the cultural significance of this bug. And Native Americans living in some rivers in Northern California actually use them as a food source during the emergence, which is typically in June every year. So not only did Native Americans eat them, but some modern Americans eat them too. Here's a
venerable BLM River Ranger Rooster Barnhart enjoying a salmonfly sandwich.
So now that you know a little bit about the bug, I'm going to talk about the river. So the Gunnison River has a long history of interesting development and influence by man. And basically that's what I want to talk about, is sort of the important dates in history and the important events in history that shape the river today. So in the early 1900s, the Gunnison Tunnel was built, and this was one of the first projects by the U.S. Bureau of Reclamation. In fact, it was one of the first 2 or 3 projects they even considered. That was built from 1905 to 1909, and it was to take irrigation water from the Gunnison River into the Uncompahgre Valley, irrigate lands around the Uncompahgre Valley that is, primarily responsible for all the agriculture in that valley today. In 1937, Taylor Park Reservoir was built, and this was built to ensure more constant flows in the Gunnison River. The Gunnison historically was a river of huge flux and of huge variability. You could have flows of 15 to 20,000 CFS one year, and the river would go dry the following year. Especially in late summer, it was not reliable as an irrigation source all the time. So the Uncompahgre Valley water users built Taylor Park Reservoir in 1937 to shore up those water supplies. The next big thing to happen was the construction of the Aspinall project. This was under the Colorado River Storage Project of 1956. It involved building 3 large dams on the Gunnison River: Blue Mesa, Morrow Point, and Crystal. So 2 of the really important things the 2 really important events that occurred on the river that really influence how the river is operated today would be the construction of Crystal Dam in 1976 and the 2012 Aspinall unit reoperation. And this reoperation basically changed how all of those dams are operated, basically to provide a more natural hydrograph for endangered fish that live downstream of the Gunnison Gorge. So the 4 native fish species that are listed under the Endangered Species Act are in peril for many reasons, but primarily because of the changes in flow and hydrograph that large dams have caused. So in 2012, they reoperated the Aspinall unit to provide a more natural hydrograph. And between that and the authorized purpose of Crystal Dam, those are really the 2 hinge pins of how the river affects specifically the fishery today. And importantly, Crystal Dam, when it was built specifically to re-regulate the flows. So Blue Mesa and Morrow Point are primarily, in addition to flood control and recreation, their primary purpose is to generate power. Well, Morrow Point Dam generates power at a peak power schedule that can go from 0 to 5,000 CFS. But they built Crystal Reservoir specifically to re-regulate those flows and basically cushion the flow changes downstream of Crystal Reservoir. So here's just a cool old-timey photograph of some of the workers that built the Gunnison Tunnel, and I just like including this because it's amazing to think about in 1905 building that 6-mile-long tunnel by hand and by mule, by dynamite. It was an amazing task in my opinion, and not insignificantly, 26 men died during the construction. It was a dangerous job as well. So this tunnel basically goes underneath, the national park, takes water from East Portal and delivers it to Montrose and Delta in the Uncompahgre Valley for irrigation. So to talk about sort of the changes in the river over time, before the early 1900s in the Gunnison Tunnel, the primary fish species in the Gunnison were native Colorado River cutthroat trout in the upper river, and then somewhere around the Black Canyon and the Gunnison Gorge, it transitioned into warm water native fish. And these native fish include the 4 endangered fish: Colorado pike minnow, razorback sucker, bony tail, and humpback chub.
Now, that exact transition from cold water fisheries of cutthroat trout to native warm water fish was dynamic in time and space. It did not occur all the time in the same place, but was probably somewhere in the Black Canyon. Now, after 1904 and 1909, when the tunnel was built, flows below the Gunnison Tunnel became a lot more intermittent, and that really changed probably where that transition occurred. And in that time period after 1909, the trout fishery below there was probably pretty spotty until Taylor Park Reservoir was built and until the Aspinall unit was built. Now, interestingly, the upper Gunnison below Blue Mesa Reservoir, before it was built, it was renowned as a trout fishery at the time. So even after non-native trout, which are rainbow trout and brown trout, replaced cutthroat trout, that wild trout fishery was excellent in the river upstream and underneath what is today Blue Mesa Reservoir. And it was also known that at the time, if you read old newspaper reports from Gunnison, how important these large emergences of salmon flies were to anglers. And in fact, there's all kinds of cool old articles and National Geographic, Outdoor Life, all identify the Gunnison River as some of the best trout fishing in the country. And the time to fish it was June, when the salmon flies were coming. So after that time period, the Aspinall project was completed in 1976. And those intermittent flows that were below the Gunnison Tunnel, so the warmer flows and the intermittent flows in the Black Canyon and in the Gunnison Gorge pretty much ended then, and that established perennial flows of cold, clean water, and the cold water sport fishery that we know today in the Black Canyon and the Gunnison Gorge was really established because of that. So all that history and all that operations really leads to a really excellent river for a lot of different things, specifically for cold water and sport fish, really robust and diverse aquatic invertebrate community. And what we have today is a river that's has base flows of about 300 CFS. I think it's probably flowing 350 right now, with peak flows anywhere from 1,000 CFS up to maybe 14,000 CFS. And these peak flows are probably less than they were historically before the dams, but are a lot more than they were before the Aspinall unit was reoperated in 2012. So what we have today is sort of augmented base flows. The dams and the operations have increased base flows. They've dampened the peak, but it is still kind of a natural peak. But importantly for trout, it's a peak with some sideboards on how quickly they go up and how quickly they go down. And that's important for survival of young trout in the river. So the other factors that really are important with the Gunnison is that there is no power peaking. Those power peak flow changes that impact many of the rivers below larger hydropower projects are ameliorated by Crystal Reservoir. So below Crystal Reservoir, we have a pretty stable hydrograph with a kind of a natural peak flow. And, this is a sort of a complicated topic. Dams have a huge effect on the rivers below them. And many impacts, both negatively and positive effects of those dams have been studied, and it's kind of too complicated to come in today, but I'll sort of summarize it by saying that the Gunnison is really unique in my opinion. It has all the benefits of a large upstream reservoir, such as the increased base flows in the cold water, and many of the negative impacts of dams, which is the reduction of peak flows and the variability from year to year has been improved by the operation and the planning, specifically of Crystal Reservoir. So like I said before, historically, of course, the Gunnison is much different than it is today. It's a judgment call to say whether it was better or whether it was worse, but of course it's different. Pre-dam flows fluctuated from virtually nothing some years to 20,000 CFS or more. So, the river is very different today, but, rivers are dynamic. they're varied and they reflect the landscapes around them, and they reflect the people that use those rivers. So today the Gunnison serves many purposes, from providing irrigation and drinking water to hydropower generation, and of course recreation in the form of boating and fishing. Luckily for us, I think that a lot of those different uses have been balanced pretty well in the Gunnison, and they result in a really cool wild river and a beautiful wilderness canyon, and honestly, one of the best trout fisheries in the Western United States. So today, the Gunnison River, as I said, supports an amazing trout fishery. It's considered gold medal by Colorado Parks and Wildlife, and it supports anywhere from 6,000 to over 10,000 wild trout per mile. And out of all the fisheries that are renowned throughout the West at having high densities of fish, there are really few rivers anywhere in the country that can hold up to the Gunnison as far as the number of wild fish per mile. It's pretty impressive. And what you see here are some large brown trout in the lower right. That's a fish, probably in the 10 to 12 pound range from the Gunnison Gorge, along with a large rainbow trout that Eric Gardino is holding in the upper left. And these are the two primary sport fish that are present today. And of course, these are exceptional specimens. These are not the average fish in the river, but the river does produce some pretty nice sized trophy fish at the top end. So flows in the river are a very hot topic, and a lot of people talk about it. A lot of people want to know, why is it this flow? Why is it that flow? How come the peak flow occurs when I want to go boating, or any number of different questions. And it's complicated. There's a lot of people that use the river, and there's a lot of agencies that have a stake in that river. First thing I'll say is primarily what affects flows in the river is Mother Nature. The amount of snow melt that we get in the Upper Gunnison Basin really drives the whole system, of course. That water is then controlled by the Uncompahgre Valley water users and the Bureau of Reclamation. The Uncompahgre Valley water users have the oldest water rights in the basin and own and operate Taylor Park Reservoir. The Bureau of Reclamation operates the Aspinall unit and the 3 large dams that occur on the river. And of course, the recreation in the river is managed by Colorado Parks and Wildlife on the fisheries end, and then of course the Gunnison Gorge, BLM, NCA on the lands end. And all of these agencies and many others that I couldn't list here have a big interest in the Gunnison and have to work cooperatively. And certainly Parks and Wildlife and the BLM have worked very closely on both fisheries and wildlife issues in the river. So now I want to talk a little bit about some of the specific research that we've done and how it relates to the Gunnison Gorge. So one thing to know about salmonflies is, while they're very numerous in the Gunnison Gorge, they're not numerous in a lot of places. And in fact, a lot of rivers throughout the West that previously had large emergences of salmonflies, they're virtually extirpated or severely reduced now. There's several rivers in Utah and Montana where declines both in range and numbers of salmonflies have been documented. And this has occurred in Colorado and some rivers as well. So because of the concerns about the loss of this important invertebrate in some of our rivers, we've undertaken several different research projects. So specifically in the Gunnison, it's an interesting story in the Gunnison because salmonflies were very numerous in Blue Mesa, in the river that was inundated by Blue Mesa and above Blue Mesa. Today, they're virtually absent from there. So here's a graph that shows the density of salmonflies going upstream to downstream from East Portal, down to Drysdale Flats near Austin, Colorado. So today, salmonflies occur in some of the tributaries in the Upper Gunnison, like Cochetopa Creek, but in the main stem Gunnison above Blue Mesa, they're absent. They're also absent in East Portal, immediately below the dam. And then as you go downstream, their numbers seem to increase. And by the time we get to the Gunnison Gorge NCA, the densities of Pteronarcys have increased substantially. And really in the heart of the canyon, in the Ute Park Reach, we see some of the highest densities of this
aquatic invertebrate that we've documented anywhere in the state, and frankly, anywhere in the country. But while we have robust populations of them in the Middle River, we have lost a significant amount of Pteronarcys habitat and Pteronarcys populations in the river above Blue Mesa. So the Gunnison is not immune to some of the range declines that have occurred in other rivers. So another example of this in our state is on the Colorado River in Middle Park. Here's another example of how a reservoir and dam operations can affect the river, and it's a very different system up there. It's a smaller system, and it's a system that is heavily diverted out of basin. So in the Gunnison River, all of the water use generally occurs within the basin. It's diverted into the Uncompahgre River, but it is essentially returns. It stays within the basin, and irrigation return flows come back into the river, around Delta. In the Colorado River, most of the water use occurs out of the basin. That water is diverted underneath the Continental Divide to support the communities on the Front Range of Denver, Loveland, Fort Collins, and all of the major population centers. So that river is a much different system, and we've seen huge declines in not only Pteronarcys numbers on the Colorado River, but we've seen a decline in many other species, and especially the diversity of mayflies, stoneflies, and caddisflies has been reduced near some of those water projects. So because of all this, we wanted to look at salmonfly habitat and try to get at some of these questions as to why maybe they declined in some places and are doing better in other places. And we wanted to focus on habitat specifically, not necessarily some of the ecological things that could be going on with salmonflies, but describe the physical habitat that supports high densities of them, and also describe the physical habitat characteristics of sites where they have been extirpated. So what we wanted to do is determine the best habitat features that explain that difference between high and low densities, and be able to provide water managers and engineers and hydrologists specific recommendations on why certain rivers, what parts of river habitat have been impacted where the salmonflies have been extirpated. So what we did was population estimates of these exuvia. And like I mentioned earlier in the talk, salmonflies crawl out of the river in June. They have this exoskeleton, which I'll call exuvia. It splits, the adult insect climbs out of the back of that, is now a flighted adult. Well, these exuvia are left all over the riparian area, both on willows and rocks and trees and anything they can crawl on. And if you get to those sites soon after emergence, you can actually count these things, use some population modeling to estimate the density, and then expand that density estimate to similar sites within that riffle. And what we basically do is we get an index to the population. So it's, of course, when we have thousands upon thousands of these insects emerging, we can't count all of them. But we can get sort of an idea of an index of the population, and we can compare site to site. And then what we did was we compared those densities to the habitat features that we were interested in. So the sites for this study were 3 rivers, the Gunnison River, of course, the Rio Grande River, and then the Colorado River in Middle Park. So we did this technique that I referred to of trying to index the population. So rather than doing traditional benthic sampling, normally when scientists want to look at benthic insects, we go out and we'll do kick net sampling or Cerber sampling of the invertebrates while they're in their larval form. And that works good for Pteronarcys, but one of the beauties of this bug is that those exoskeletons are so easy to find and they're so easy to identify. Benthic samples, you have to have trained people that know how to identify the insects, pick all the samples, and it's pretty labor intensive. So what we did was we came up with this technique of using population modeling that's a lot of times applied to trout and to
terrestrial animals, and we applied it to the exuvia of the Pteronarcys salmonflies. And what this gave us was an easy way to estimate their densities and then compare the densities from site to site. So then we had habitat variables that we thought would affect stonefly densities, and we came to these conclusions based on literature review and then discussions amongst ourselves and with other collaborators who have studied salmonfly habitat. And what we looked at was width to depth ratio, slope, and then substrate size. So how big is the cobble in the riffle sites, the amount of fine sediment in the riffles, and the amount of cobble embeddedness. And what embeddedness is how cemented are those cobbles into the riverbed by fine sediment. So in addition to measuring the habitat specifically, we also used an acoustic Doppler current profiler to basically map all of our riffle sites. So this is a collaborator, Eric Richer, who's a hydrologist, and he's pulling an acoustic Doppler profiler that is essentially creating a digital map of the riverbed below that boogie board looking thing. So this is sort of what the survey gives us, and then from the survey points and the depths and the velocities that are measured, we can create basically a topographical map. And importantly, not only do we get a topographical map of each site, but it gives us water velocity information as well. So we can estimate things like the average velocity, the maximum velocity, the depths, all of the things that affect trout and affect the habitat that aquatic invertebrates are going to live in can be estimated by this technique. So in addition to the physical, the mapping of those sites, we measured fine sediment and embeddedness. And what you can see here is a cobble that we've pulled from a riffle site, and that light area on the bottom of the cobble is the amount of that cobble that was buried in the sediment. And by measuring the distance that it was buried and doing this a couple thousand times per site, we get an idea of the average level of cobble embeddedness for that riffle site. So the results of this study, I won't go too much into details, but basically what you want to take away from this is out of those variables that we looked at, fine sediment was sort of the winner. Fine sediment was by far the best predictor of salmonfly densities. And in this left column, you'll see just the correlation. And so the higher numbers are better, they mean a stronger correlation. Negative numbers mean a negative correlation. So as that variable goes up, salmonflies go down. A positive number means a positive correlation. So like fine sediment was our best predictor. The D50, which is the average size of the cobble particles, and embeddedness was our second place predictors. And then width to depth ratio was kind of the third predictor. So after some statistical modeling and some smoke and mirrors and hand waving, we came up with some regression models that were able to predict salmonfly density based on these different characteristics. And what you see here is sort of the results of all that modeling. And what the takeaway here is just generally how those variables change in the different sites. So from the top to the bottom, you go from high densities to low densities. So in the bottom row, what you see is the maximum densities observed. So places like Ute Park. And what you see in places like that is very little fine sediment, very large average size of cobbles, and very little embeddedness. And those variables change over space and time, across these different densities. So, as percent fine sediment increases, the density of stoneflies goes down. As the average cobble size decreases, gets smaller, the density of stoneflies went down. And as the amount of embeddedness increased, the density of stoneflies go down. So what we learned from that basically is that salmonflies are associated with cold water habitat that had very low embeddedness, large median particle size, and a low width to depth ratio with very little fine sediment. So basically riffle sites that had large cobble, maybe 6 inches and bigger, and had very little fine sediment, were the best sites in Colorado for high densities of Pteronarcys stem flies. So what does all this mean? And what can we do about it, basically? So these habitat variables that we measured, they're all affected by flow, and they're affected by the land use surrounding the rivers. So, flow regime, the sediment supply, and many other variables that are controlled by things like reservoirs and irrigation withdrawals affect that habitat. So if you want to reduce fine sediment in a river, you might look at the inputs of fine sediment. So land management. So what's the grazing? What's the logging? What's the, things that might cause erosion, things like that. And alternatively, not just from the sediment supply standpoint, but flow regime of the river really affects how fine sediment is distributed in the river. So if you have higher base flows or bankful flows, generally that keeps fine sediment from accumulating in riffles. We also know that salmon flies like large cobble sizes, and the cobble sizes in riffles is directly related to sort of peak flows generally, and that's kind of an overly simplistic way of putting it. But the higher peak flows, the average annual peak flows you have, the larger those cobble sizes are going to be in the river. So these are things that water managers and Bureau of Reclamation can frankly control. The base flows in the river and peak flows in the river can be controlled by large dams. So here's some examples of what you might want to do to a river if you want to improve these habitat conditions. So kind of to revisit the Gunnison River and bring this back and how it applies, the Gunnison is a really interesting river in this research. So as I talked about, fine sediment and some of the land use practices that may increase fine sediment, that's not really applicable to the Gunnison as much as it is to the Rio Grande or to the Colorado River. In the Gunnison, as you guys, most of you know, it's a highly erosive system. We have the upper layers of the Gunnison Gorge are dominated by that Manka shale and that really fine, highly erodible sediment. And that's not necessarily an artificial or an unnatural system. That's exactly, it's a natural state there. And that fine sediment gets washed into the river through, thunderstorms and our monsoon storms in the summer, which if anyone is on the river this summer, of course, they saw a lot of. And these huge fine sediment deposits you would think would cause, pretty poor Pteronarcys habitat. But see, that's the interplay between the land use and the landforms and the flow regime of the river. What the Gunnison has is not only augmented base flows, but it has a pretty natural peak flow. We'll see peak flows in the Gunnison anywhere from 6,000 to 10,000 CFS. Under the current operation, the Gunnison Gorge rarely goes over about 12,000 CFS because of flooding that would occur downstream in Delta. But those high peak flows do a good job of moving that fine sediment out of the riffle areas. And we see a lot of fine sediment accumulation, but it's generally within the pools and some of the flatter water in the Gunnison Gorge. The riffle habitat itself is remarkably free of fine sediment and provides excellent habitat for a lot of aquatic invertebrates, including salmon flies. And this is reflected in that sort of change in density that I discussed earlier. The habitat that's provided in the interior gorge has a lot to do with that flow regime and the natural geology. As we get closer to Crystal Dam, the density of salmon flies goes down, and that is not necessarily a physical habitat thing. That we think is related to temperature. So here's an example of how the altered flow and temperature regime of a large impoundment can negatively affect an invertebrate. If you remember earlier in the talk, I mentioned that the eggs of salmon flies stay in the sediment for 9 to 10 months. Those eggs need cold temperatures to come out of that diapause. So like right below a dam, classic impacts of a large impoundment are colder water in the summer and warmer water in the winter. So as you get close to the dam, we don't get those freezing temperatures. And we think, we're looking into this now, but we think that those eggs never come out of dive-wells. And as you go farther downstream and the temperature regime becomes more and more natural, then we see a lot more larval salmon flies. So you'll see salmon flies in East Portal, you'll see adults, but they're adults that have flown from upriver. You very rarely see larval salmon flies anywhere, much above, Warner Point, Gunnison Point area. As you get closer to East Portal, it's really just the adults that you see. So that's pretty much it. I'm not sure how many questions we have. I did want to just quickly say that I want to thank some of my co-authors, Eric Richer with Colorado Parks and Wildlife, and retired CPW researcher Barry Nearing had a lot to do with some of this research, as did all kinds of people that helped with the bug picking, the analysis, and some of our collaborators in Montana and Utah. I also want to thank the great personnel with the BLM, specifically the Gunnison Gorge NCA. A lot of this cooperative work could not have been done without their help, both collaborating at a sort of a management level, but also getting down into the weeds and helping us get boats through the Gunnison Gorge and pick bugs. So thanks to them, and thanks to all the people that have contributed to this research over the years. So that's pretty much it. I'll stop sharing my screen, and I think we have some time for questions. Okay.
Awesome. Yeah. So if anybody has questions, please type those up in Q&A or in the chat, and we have some time to answer them. Thanks, Dan. That was really fascinating. The acoustic Doppler boogie boards really caught my attention.
Yeah, we call it a boogie board, but that's I don't know the exact cost, but that boogie board probably cost about $25,000. So it's not a boogie board, but it's a pretty cool tool. It's a neat way to not only measure the river, but it allows us to measure flows in areas where you can't wade too. It's a pretty slick way of getting discharge measurements in places that you can't wade.
Yeah, looks like Joel started to type something maybe, but we don't have a full question.
Okay.
Okay, here we go. You mentioned other Western rivers have seen declines. Why there and not the Gunnison?
That's $1 million question. I'm collaborating right now with a researcher out of University of Montana in Missoula, and we're specifically looking at that. So that habitat study that I spent the last quarter of the talk talking about, we're using similar techniques and expanding the variables that we're looking at to things like temperature and flow, and some of the biological characteristics that are a little harder to measure. And we are now collaborating with them to do these measurements on several rivers in Utah, and hopefully we're going to be able to figure that out. And unfortunately, I mean, it's not going to be a simple answer. Nothing in ecology ever is a simple answer, and each one of those rivers has a specific suite of influences and changes over time why the salmon flies might have gone away. But there's also going to be probably some similarities between those systems. So some of the rivers in Montana and Utah that have seen salmon flies declines, they also have large impoundments on them. But those impoundments are operated very differently than the Aspinall unit is operated. So we think that has a lot to do with it. But of course, there's, each river is going to be its own. There's some rivers, frankly, that are impacted by things like metal pollution, where it's a pretty easy answer that, places that heavy metals have been introduced into the system, salmon flies get removed pretty quickly. And, that's probably, frankly, what happened on the Arkansas and parts of the Upper Gunnison, even in areas that had a lot of mining history. We don't see a lot of salmon flies. So that's a long answer, but it's probably going to be different from all the different rivers, but we're hoping to figure that out. And within the next couple of years, we might have some answers.
Very interesting. We have a couple questions about the revival of salmonflies above Blue Mesa. So someone references relocation efforts of nymphs to the upper Gunnison area. Is that working? Any insights on that?
That's been tried, to my knowledge, probably 4 or 5 times, and it's never worked. And there's lots of debate and conjecture as to why it doesn't work. But it's, I mean, as recently as, I don't know, maybe 5 or 6 years ago, there was a group in Gunnison that was attempting a sort of a relocation effort. And, you know, that's probably a complicated question. The upper Gunnison has changed remarkably from when there was lots of salmonflies there. The temperature regime is different because of Taylor Park Reservoir. And frankly, the lower end of the river is vastly different because of Blue Mesa Reservoir. And then the third factor I think I'd put in there would be heavy metals, you know, some of the mining impact that comes out of the East River and the Crested Butte area. How those 3 factors are affecting the salmonfly habitat, we don't know exactly. We are doing measurements up there with this expanded study that I talked about with our collaborators from Montana. And we're going to try to look at some of that and maybe get some answers as to why salmonflies are pretty much gone from the main stem river. It may be as simple as that was always their sort of their upstream distribution. And once Blue Mesa flooded out the prime salmonfly habitat, you know, around Savoia, around Iola, excuse me, you know, that the upper river was just less, you know, quality habitat, and that combined with the altered temperature regime that comes out of Taylor Park Reservoir might have been enough to sort of reduce the habitat up there. But anyways, we don't know specifically. We know it hasn't worked. Hopefully we'll have some answers in some future, but I can confidently say that salmonflies are not in the upper Gunnison because they haven't been put there. They're not there because it's not suitable habitat for them right now.
Gotcha. One kind of just background question. What other parts of the country are salmonflies found in?
Yeah, they're found all over. You know, and there's a couple of closely related species that occur in other parts of the country, but our species, Pteronarcys californica, are found throughout the Colorado River Basin and up into the Pacific Northwest. So you hear about them. There's rivers in Northern California that have high densities of them. The Deschutes River in Oregon is known for having high densities of them. They occur in Washington on the Olympic Peninsula, and of course all across Idaho and Montana. You know, rivers like the Henry's Fork, the Madison, the Big Hole River in Montana. So they're pretty much all over the West where there's good habitat.
Yeah, we have a couple questions relating to the snowpack. So is the flow rate timed with expected peak hatch time? The biggest hatch I have seen, says Jason, was in 2016 with the very high snowpack. And there's another kind of similar question of, does snowpack influence the year-over-year population numbers?
That's a complicated question, and we probably have another hour-long discussion to snowpack influences everything in the river, right? I mean, the flow regime from year to year changes, and it's based pretty much directly on snowpack. Because these bugs live in the river for 4 years, they have 4 years to be impacted by things like drought and high flow years. So it's a little bit hard to make a specific explanation as to why it was a great year in 2016 or something like that. To bring it to this discussion, those high flow years are very important in maintaining that habitat. So like a lot of anglers gripe about high flow years because during those high flow years, the peak flows occur during the emergence and it makes it harder to fish. Frankly, if we didn't have those high peak years, we wouldn't have the quality habitat and support those high densities of Pteronarcys. So the fishing may be terrific during a sort of a moderate flow year of like 2000 CFS peak. But if you had that repeatedly for 20 years, the salmonfly population would probably decline. As far as like, are high flows always good for them? It certainly supports their habitat. You know, the annual emergence, like why one emergence in 1 year is bigger than the next emergence, I can't answer that. There's a lot of different factors. And again, it's probably
an accumulation of factors that occur over that 3 years where there are nymphs crawling around in the river. But yeah, we see big emergences during high flow years. I've seen some pretty big emergences during low flow years, and that variability from year to year can be pretty extreme. But yeah, so the fast answer is that snowpack affects everything in the flow regime, and therefore affects all life stages and all aspects of salmon fly habitat.
Yeah, that seems like a lot more research could be done on that topic. Next question from Kara. Given how compelling the results of your study are, how successful have you and your fellow researchers been in persuading other rivers to employ some remediation tactics?
I would say partially successful. The one spot I can point to, and one of the driving forces behind this research was the Upper Colorado River, which I mentioned during the talk. Like, we've seen massive declines there. Because Denver Water and Northern Water are the two big partners that control most of the trans-basin diversions up there, they've proposed some massive changes to their system that's going to increase diversions from the Upper Colorado River. So as mitigation for that increased diversions, we've, along with other partners and agencies, have gotten some concessions and some mitigation efforts specifically around Windy Gap Reservoir. So Windy Gap Reservoir is the reservoir I showed in that one graph where below that reservoir, the salmon flies have virtually, been eliminated. They're still there, but they're in very low densities, and they're not certainly in the densities that they were historically. So that reservoir specifically is being taken offline. A channel is being built around the reservoir to reconnect it. And so those diversions will now occur off channel, which we think will have a huge beneficial impact to the ecology of the river and improve salmon fly habitat. So that's probably the one success story I can point to where we think this research is being applied. We hope maybe in the future we'll have some more success stories to tell you about, but that's the only one I can think of right now.
Yeah. Okay, we have another question that sort of relates to what you were talking about towards the end in terms of dams. So how much does temperature affect population? Dams moderate temperature extremes, but lower water means higher temps, especially the further downstream it is. So does the current drought cause concern for salmon fly populations?
That too is a complicated question. And of course, if you ask an ecologist anything, they always answer, it depends.
Yeah.
And of course, it depends. And so, dams, it depends how big they are, and it depends how big they are in relation to the river. So the general pattern that we see is that the cold water from dams is not necessarily a negative thing. The warmer than average temperatures right below a dam, and I should put some context to that. I mean, warmer than it would have been without the dam. So, below a lot of dams, there's no ice anymore for some distance. And so in those zones where we don't see freezing temperatures, salmon fly populations have probably declined. And then as you get farther away from the dam, sort of the benefits of the cold water are seen in that habitat, and they probably help salmon fly numbers, But of course, it's different for every system. The Madison River has seen some declines in salmon flies. That's a much different system than we see in the Gunnison. So how a drought is going to impact those, is probably going to vary by rivers. I would say, it's hard to make a generalization about that. In the Gunnison itself, I'm not worried about it. The operations plan and the amount of storage that are in that system, future habitat is probably going to be fine to support very high densities of salmon flies. Some rivers like the North Platte in North Park and the Rio Grande River, I think are going to be much more susceptible to, if we see decreased flows and warmer temperatures because of drought, the lower end of salmon fly distribution is probably going to be contracted. So, the upper end of salmon fly distribution is maybe influenced by the dams, but as temperatures warm up and flows go down, it's the lower end of the distribution that is going to be impacted. So I think, if I had to guess, rivers like the Colorado River,
the Rio Grande and the North Platte, you could see some declines in the future with warming temperatures and lower flows.
Yeah, I love it's complicated because really, right, there are so many factors. Clearly from your research, you're already looking into a handful, but the ecosystem provides that many more. I think unless anybody else has additional questions that they can type up really quick, that about concludes our time. So we do have one more lecture happening 2 weeks from now. That one is about pack rats and how they create time capsules with things they find and their urine. So that's going to be another really interesting kind of ecology focused lecture. Like, let's see. We're just getting some thank yous now. So I think we'll end it here. Thank you everyone for attending. Thank you, Dan, for all of that information and for going into all of the details of your research and sharing that with us. I know I learned a lot. And yeah, we appreciate your time. Thank you everyone for joining us. And I hope you can continue to attend CCA events and lectures and all the various things we put on.
Thanks, Annie.
Thank you. All right.